and the production of genetically unique gametes. So we're going to go through some of the mechanisms of meiosis, and we know it's two separate divisions, Meiosis One and Meiosis Two. We know the parent cell, in this case, is going to be a diploid cell, and we know the four genetically unique daughter cells that we get at the very end of Meiosis Two are going to be haploid, and we do have to use the term genetically unique. Very often they are gametes. So let's start with our parent cell, which is the top left. Now, in the human body, this might be one of the cells in the testes that will form the sperm cells eventually, or it could be one of the germline cells in the lining of the ovary that will undergo meiosis to form, the immature egg cells. So at the top, we can see the note says before S phase. So if this cell had, for example, 4 chromosomes, we can see, hopefully from the diagram, from the red chromosomes that I've just drawn in, that there's 2 big red chromosomes and 2 smaller red chromosomes. So we can see there's 4 chromosomes, and each chromosome is a single molecule of DNA. The large chromosomes, there's 2 of them, and they look the same size. The smaller chromosomes, again, there's 2 of them, and they look about the same size. So we've got 2 sets of chromosomes here. We've got one big chromosome on the left, and that might be the maternal chromosome. We've got an equal length large chromosome just next to that, which might be the matching chromosome from the dad. The third chromosome is a smaller chromosome, that might be from mom, and the matching smaller chromosome might be from dad. So there's 2 sets of chromosomes here. So the ploidy we are going to describe as diploid. Diploid simply means 2 sets of chromosomes. Now we're going to use an arbitrary unit for DNA mass. So in this case, we're just going to say the mass is 4. Now the arbitrary unit's a relative unit that allows us to kind of look how that DNA mass is going to alter through cell division. Now we know S phase is a part of interphase where DNA replication takes place. Now DNA replicates via a mechanism called semi-conservative DNA replication. So on the very left-hand side, we can see there's 4 molecules of DNA, or 4 chromosomes. Each chromosome is a single molecule of DNA. However, after S phase, so after S phase, we know that each molecule of DNA is replicated. So there's going to be 2 molecules of DNA which are joined at the centromere. So after S phase, this original molecule of DNA has replicated, and we're going to see that is linked to the identical molecule of DNA that was produced from replication. That is a single chromosome. It's a replicated chromosome. The second molecule of DNA, so this one here on the left, that will also have replicated. So we're going to get 2 identical molecules of DNA joined at the centromere. That's the second chromosome, which in this case in the center is replicated. The first small molecule of DNA on the left, I'll just put a tick next to it. That small molecule of DNA is also replicated after S phase. So there's going to be 2 identical molecules of DNA joined at the centromere. And the 4th chromosome on the left, so I'll put a tick next to that. That might be the paternal molecule of DNA, the paternal chromosome there. That is also replicated. So each of the original molecules of DNA have replicated. There's 2 identical molecules of DNA that are joined at the centromere, which will form each individual chromosome. So a replicated chromosome is made from 2 sister chromatids joined at the centromere. Now the replicated chromosome is still classed as a single chromosome. So on the left-hand side, we had 1, 2, 3, 4 chromosomes. On the diagram in the middle, we've still got 1, 2, 3, 4 chromosomes. The only thing that's changed is that each chromosome in the middle of our diagram is now a replicated chromosome made of two sister chromatids. So the ploidy, we've still got our 4 chromosomes. The ploidy is diploid, so it stays the same after S phase. However, the DNA mass has doubled because each chromosome is now made from 2 sister chromatids joined at the centromere. The sister chromatids that make up a replicated chromosome are identical molecules of DNA, and they'll have exactly the same alleles along the length of the sister chromatids that make up that replicated chromosome. Okay, now prophase 1, we know the nucleus would disintegrate and the nucleolus would disappear. The first 2 diagrams on the left and in the middle haven't shown the nucleus, but at this stage they would be inside the nucleus. So the middle diagram, these 4 replicated chromosomes would be chromatin. So we could say chromatin here, and they would be contained within the nucleus. Prophase 1 on the right is the first stage of meiosis 1. And we know after the nuclear envelope disappears and the nucleolus goes, the chromatin supercondenses. So these replicated chromosomes are now visible under a microscope if they've been stained with a particular dye. Now, what you're going to see is each replicated chromosome, they're visible because the chromatin has supercoiled. So the long DNA molecules that make up the sister chromatids of each chromosome are shorter and fatter after supercondensing. So we'll have the same 4 chromosomes, but the sister chromatids look shorter and a bit fatter, so they're visible. So we've still got the same 4 replicated chromosomes. Here we go. But because the chromatin has supercoiled, the DNA is wrapped around those histone proteins nice and tightly. Each replicated chromosome is now visible under a microscope with a suitable stain or dye. So here in prophase 1, we can see the ploidy is still diploid. There's 2 sets of chromosomes. There's 2 versions of the big chromosome. One might be maternal and one might be paternal. And the small chromosome, again, one is maternal and one is paternal. So there's 2 sets of chromosomes. So the ploidy is diploid, and we know the DNA mass remains at 8 at this stage. So this would be prophase 1 of meiosis 1. And we can see those nice supercondensed replicated chromosomes. Now, meiosis 1 then, if that was allowed to continue, so it goes PMAT, so prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. So in prophase 1, these replicated chromosomes, the homologous chromosomes, would actually pair up into a bivalent. So the maternal chromosome, replicated 1 at the top, would pair up with the paternal replicated chromosome into a bivalent, and the smaller chromosome, the maternal one, would pair up with its matching homologous chromosome from the dad, and we get another bivalent. So this cell in the top right, we'd end up with 2 bivalents. Okay, and that would be the end of prophase 1. And those bivalents, the 2 bivalents, would attach to separate spindle fibers, and they'd line up down the equator of the cell, and that would be metaphase 1. Now in anaphase 1, we know the homologous chromosomes separate and they move to opposite poles of the cell. So the bivalent, the 2 bivalents, the 2 homologous chromosomes that make up the bivalent, they separate to opposite poles of the cell. And we'll talk about independent segregation shortly. And that's a source of genetic variability in meiosis 1. So at the end of meiosis 1 then, after we had cytokinesis, what we'd end up with is one of the big replicated chromosomes and one of the small replicated chromosomes. And the other daughter cell would be the same. So we'd have one of the big replicated chromosomes and one of the small replicated chromosomes, because the bivalent separate the homologous chromosomes that made up the bivalent, separated to opposite poles of the cell, and then we had division of the cytoplasm to create the two daughter cells. So if we look at this cell from the end of meiosis 1 on the left, we can see there's that chromosome 1, and there would have been maybe the chromosome 3 that we had earlier. And on the right hand side, the second cell on the right, we can see this is the large chromosome that we call chromosome 2, and then we've got another small chromosome we call chromosome 4. So each daughter cell then at the end of meiosis 1, we can see there's two replicated chromosomes, one here and one here. So this time there aren't matching chromosomes. There's a single replicated chromosome. So there's only one set of chromosomes. So the ploidy is what we call haploid, and that's a single N. So we've gone from two sets of chromosomes at the top to only a single set of chromosomes at the end of meiosis 1. Now, if we look at the DNA mass, then hopefully you can see that the DNA mass is halved from that original cell after S phase. So we saw up here in the middle, okay, this was after S phase here, the DNA mass was 8 arbitrary units. So we could actually count sort of 8 strands of DNA or 8 molecules of DNA. Here at the bottom on the left, the daughter cell, we've got a DNA mass of 4. So not only have we got half the number of chromosomes, we've also got half the DNA mass compared to after S phase at the top. Now this DNA mass, if we look now, actually matches the original parent cell before S phase. So the second daughter cell on the right hand side, then we can see one large replicated chromosome, and we can see a small replicated chromosome. There's only one set of chromosomes here. There's the big one and the small one. There isn't the matching homologous chromosome. That's in the other cell. So again, the ploidy is haploid, and the DNA mass again is 4. So that's meiosis 1. Now, if we took these two cells at the bottom and we allowed them to divide by meiosis 2, we'll see what happens next. So imagine we had the same two cells, and then we'll draw them in here. So this is one chromosome that I've just drawn. And then we had the smaller chromosome with its two sister chromatids. And we had the same scenario on the right. So we know these are haploid, and we know the DNA mass is 4. We're going to get meiosis 2 this time, which is very similar to mitosis. So the process would be prophase 2, metaphase 2, anaphase 2, and telophase 2. We then get cytokinesis to form our 4 genetically unique gametes. Now we know meiosis 2, because it is similar to mitosis, that each replicated chromosome would line up on the equator attached to a separate spindle fiber. So we might have this arrangement here on the left, attached to separate spindle fibers. And we know that the spindle fibers contract, and that separates or splits the centromere, and it allows the sister chromatids to be pulled to opposite poles of the cell in a V shape. And eventually we'll get telophase 2 and then cytokinesis. So what you'll end up with is one of the large sister chromatids from the original replicated chromosome, and you'll get a small sister chromatid from the other small replicated chromosome. We'd have the same in the second daughter cell. So one large sister chromatid from the large replicated chromosome at the top, and one small sister chromatid from the small replicated chromosome. We'd have the same actually for the second daughter cell from meiosis 2, which is this one on the top right. We know the centromere would split during anaphase 2, and we get one large sister chromatid there, and we get one small sister chromatid from the other replicated chromosome, and it'd be exactly the same for the 4th gamete here. So we can see each gamete has one large sister chromatid from the first chromosome, and it has a small sister chromatid from the second replicated chromosome. So at the end of meiosis 2, we can see actually that the ploidy is still haploid. It's just at this time there's one set of chromosomes, which actually they're chromatids, but we can now call them chromosomes again. The cells at the top, there are 2 chromosomes in each cell. Okay, one big one and one small one, but they're replicated chromosomes. They're still classed as single chromosomes though. So these cells at the top we know are haploid. The genetically unique gametes are also haploid, but the DNA mass this time is halved, so it's gone from 4 to 2. Now the second gamete, we can see the ploidy. There's one set of chromosomes here. The ploidy is haploid, is N. DNA mass is also 2. Same for the third gamete, and the same for the fourth gamete. So actually the DNA mass is half of the original parent cell, the diploid parent cell, before S phase. Because the original DNA mass was 4, and here in the gametes the DNA mass is 2, which is half of the original parent cell before S phase. The ploidy of the gametes is haploid, just like the ploidy of the cells at the end of meiosis 1 is haploid. The original parent cell, the germline cell, had a diploid number of chromosomes. Okay, so it had 2 sets of chromosomes. So that's meiosis, and that's how we get genetically unique haploid gametes. Now, I'm going to do the same thing again, but this time we're going to introduce a process called crossing over. So from the first meiosis 1 that we saw a minute ago, we know that independent segregation of homologous chromosomes from each bivalent is a random event, and it produces random shuffling of chromosomes to either daughter cell. And that introduces some genetic variability, because the bivalent can attach one way or it can attach completely the opposite way, and that's random, and it's separate from all the other bivalents, and that's called independent segregation. But also we know that we get crossing over between chromatids of the homologous chromosomes in a bivalent. So let's do this again, but we'll try and show or demonstrate a little bit of crossing over. So the parent cell we know is diploid. We'll use the same DNA mass of 4, which is an arbitrary unit, and we'll have 2 big chromosomes, so 2 big molecules of DNA, one from mum, maternal, one from dad, paternal. The second type of chromosome is going to be a bit smaller, just like we had earlier, so that's 1 molecule of DNA, maternal. The homologous chromosome is just 1 molecule of DNA, but it's from dad, it's paternal. We've got 4 chromosomes here. We've got 2 big ones, so 2 sets, and we've got 2 little ones, so the DNA mass is 4. Now, after S phase, we know the first molecule of DNA is replicated, and it's going to be joined at the centromere. So we've got 2 sister chromatids here, which are linked to the centromere. Obviously, this is the maternal big chromosome. This is arranged as chromatin, so we won't be able to see it down a microscope. It's not visible just yet. The second homologous chromosome, which is the same size as the first one, that is also replicated. So that's made of 2 sister chromatids joined at the centromere. The small chromosome, that molecule of DNA, that's also replicated. So we've got 2 identical molecules of DNA. So that's now a replicated chromosome. Let's do that again. There you go. Maternal. The 4th molecule of DNA gets replicated. That's now made of 2 identical molecules of DNA called sister chromatids joined at the centromere. So we know here the ploidy is still diploid, but we know the DNA mass after S phase is 8, because the DNA mass is doubled through DNA replication. Now we know that these replicated chromosomes are going to supercondense and coil and become visible in prophase 1. So we might draw these in. Let's have a look. There we go. Now I'm going to draw these as bivalents. So I'm going to put the second big chromosome next to the first big chromosome. They're both replicated. So that process is called synapsis. where the replicated homologous chromosomes pair up. So this is chromosome number 1, and this is the second chromosome. We'll call that number 2. They are the same size with the same genes at the same loci. They're going to pair up. The other small chromosome that's replicated is going to pair up with the other small chromosomes. That's maternal, and then that's paternal number 4. Now, I'm just going to draw this one in red here. So just to show that this is the homologous chromosome, but it's maybe the paternal chromosome. So it's the same size as the one on the left in white, but this is the red one. Now I'll do the little chromosome that's replicated in red as well. So it's got its 2 sister chromatids in red. There you go. And that's homologous to the other white small chromosome. Now I'm just going to put here what would happen underneath if we had crossing over. So I'm just going to redraw this. So I'll do the sister chromatids in red. Here we go. But this time what I'm going to do is just draw on where crossing over has taken place. So crossing over, if you can just see that little part of the chromatid, crossing over is where non-sister chromatids form a physical link and an equal length of the chromatid is swapped over. So this white chromosome that's replicated on the left, that we said was chromosome 1, now has a little segment of the chromatid from the homologous chromosome. Now the red replicated chromosome that we called number 2 has a little segment of white chromatid that was originally part of the other homologous chromosome. So that's swapping over. It's called crossing over a part of the chromatid. Now we might also get that with the small chromosome. So I'm just going to draw this here. Okay, so these are the bivalent. This is the bivalent. And you've got the paternal chromosome in red. There you go. And I'm just going to draw a little segment at the bottom here. So this would have a white bit at the end, like that. So we've also got crossing over from the other bivalent. Now, if I drew what these daughter cells would look like at the end of meiosis 1, we would have on the left-hand cell the large chromosome that's replicated, the white one, maybe the maternal one, with the little segment of the other homologous chromosome in red. And we might have the smaller chromosome in white like this, again with its little swapped over part of the chromatid. So if that was the case, in the other daughter cell on the other side, we'd have the red homologous chromosome, but with the little white bit of the chromatid at the end, if you can see that at the tip here. So that little bit at the end, this bit here, was the part of the chromatid, the equal length of the chromatid from the white homologous chromosome. Now we'd have the little chromosome in red too, and I'm just going to draw the little white bit at the end, if you guys can see that. So you've got your 2 sister chromatids joined at the centromere, but hopefully you can see the swapped over part. So if I just put all the arrows to show the swapped over piece of chromatid, hopefully you can now see it. So the white homologous chromosome, the big one on the left, has a little piece of red, which is part of the matching homologous chromosome that we had earlier in red. And then the small white chromosome, so we'll go chromosome, what do we call them? Chromosome 1 here, yeah. This was chromosome 3, this was chromosome 2, and this was chromosome 4. So you can see the little swapped over bits of chromatid. Now, if we take these 2 cells at the bottom, these 2 daughter cells, and we draw these out again, we can take this process through to the end of meiosis. So we're going to put in that chromosome there with its little sister chromatid with a little part of the red chromatid at the bottom. We have the smaller chromosome again with a little piece of red. The other cell from the end of meiosis one looked like this. So this was the paternal chromosome maybe. So we've got two identical molecules of DNA apart from the little bit at the end. And then we had the homologous small chromosome like this, with the white bit swapped over at the end. Again, this is made from two identical molecules of DNA apart from the little segment at the end. So if we go through all the processes of meiosis 2 then, so prophase 2, metaphase 2, anaphase 2, and telophase 2, and then cytokinesis, what we're going to see is that we know this time in meiosis 2, the citrochromatids split. So let's split the citrochromatids this time. So we're going to have that large white cistrocromatid in the cell on the left, and we might have the white cistrocromatid from the smaller replicated chromosome. That would mean in the second cell we'd have the other large cistrocromatid, but with the little red swapped piece of DNA at the end that was from the paternal homologous chromosome. We'd have the smaller chromosome also with a little bit of crossed over DNA from the paternal one. The third daughter cell, the third gamete, we would have the large cistrocromatid in red, but with the little piece of white swapped over DNA at the bottom, crossing over. We'd have the small cistrocromatid from the other chromosome with its little piece of swapped over DNA. And then lastly, we'd have the cistrocromatid that was all red. So this is all the maternal DNA from that large chromosome. And then we'd have the other chromosome just in red, which was the smaller chromosome. Like that. So if you look at the ploidy of the cells at the top, well, we know they're haploid because there's only one set of chromosomes, one big one, one small one. The DNA mass is 4. The gametes that are produced at the end of meiosis 2, okay, so the end of meiosis 2, the ploidy again is haploid. So we can see there's only one chromosome of each type, or one chromatid of each type now. And the DNA mass is half of that of the cells at the end of meiosis 1. So the DNA mass is 2 for each gamete. Now, if you look at gametes 1, 2, 3, and 4, you can see they're all genetically unique. So gamete number 1 is different genetically from gamete number 2, which is genetically different from gamete number 3, which is genetically unique and independent of gamete number 4. So that's what we mean by genetically unique gametes. And we know the gametes in this case are haploid. So meiosis then has got two divisions, meiosis 1 and meiosis 2. The parent cell was diploid and the gametes are then haploid. So meiosis halves the chromosome number from diploid to haploid, from 2 sets of chromosomes to 1 set of chromosome. So if this was in humans, we know we've got 46 chromosomes in the diploid parent cell, and we know our gametes, whether it be sperm in males or eggs in females, would have 23 chromosomes, or 23 chromatids, which we now call chromosomes again. So let's just take a quick look at an exam question. Now this question says, at the end of meiosis, okay, so we know it's about meiosis, produces haploid cells called spores. So these are not gametes as such, but they're spores, which are haploid because they've been made from a diploid cell through meiosis. So we know the parent cell would be diploid, and these spores are going to be haploid. That means they have 1 set of chromosomes. Then it says the spores are produced in narrow tubes that restrict their movement. So as a result, each tube contains a single line of spores. So it's basically saying they've got little like capillary tubes, and the spores get stuck inside here. So we're going to see in a minute that each tube can fit 4 spores in it. Now it says the spores are either coloured pink or white. So the spore colour gene, so that's one gene at a given locus. So locus just means position of the gene on the chromosome, is located on a pair of homologous chromosomes. Each zygote produced in this cross has 1 chromosome with a pink allele, and one chromosome with a white allele. So this zygote then, we actually know is diploid. So this would be the first homologous chromosome and it's replicated, and this would be the matching homologous chromosome. So same size, you can see there, same genes, so it's the gene for spore colour, and the only difference is that the alleles on the left hand side for the first homologous chromosome are the pink allele, and the matching homologous chromosome on the right, number 2 here, instead of the pink allele, it's got the white allele for spore colour. So the gene is for the colour of the spore, and the allele is the variant of the gene that gives the phenotype. So here, the gene for spore colour is pink on the left, number 1, and it's white on the right hand side for number 2. Now we know this zygote, okay, it's after S phase, because each chromosome, we just have one chromosome, that is now made of 2 molecules of DNA. The other chromosome is also replicated, and that's made of 2 molecules of DNA. So this one on the left would be the one with the pink allele, so the pink allele would be here, and the homologous chromosome on the right would have the white allele. Then it says the first nuclear division in meiosis produces these 2 cells underneath. So we can see there's 1 homologous chromosome in the cell on the left and 1 homologous chromosome in the cell on the right. So we'll put number 1 there, number 2 there. So we've gone from 2 sets of chromosomes at the top to 1 set of chromosome in these 2 cells. So we know these must be haploid because it's the end of meiosis 1. We know the chromosome on the left that I put number 1 above, it's still replicated. It's made from 2 identical sister chromatids joined at the centromere. So the centromere would be just here, but it's still classed as a single chromosome, even though it's made of 2 molecules of DNA. Now, the second nuclear division, we know, if I just draw this out, 1, 2, 3, 4 cells. So this cell over on the left here would form 2 cells, and the sister chromatid would be pulled apart as the centromere split. So we'd have that sister chromatid in the first cell, and we'd have the other identical sister chromatid in the second cell. The third cell, we'd have 1 sister chromatid with the white allele, as we would do in the 4th cell. So this cell I've just highlighted here, if you can see that one, would split to make these 2 gametes on the right, which are the white alleles. So spore number 1 would have a pink allele. So that is, do that again, pink. Spore number 2 also has a pink allele. That would be pink. Spore number 3 has a white allele. That would be white. And spore number 4 has a white allele. So that will also be white. So we've collected 4 spores, or 4 haploid cells at the end of meiosis, and we've collected them in that tube. The first 2 spores on the left have the pink allele, and the 2 spores on the right hand side have the white allele. Okay, so the first question then, it said there are 7 chromosomes in a spore nucleus. So we need to highlight that, and we need to highlight spore nucleus. It says place a tick in the box next to the number that represents the number of chromatids present in the zygote. So we've got to pay careful attention to what the question's given us, right? So we know the zygote is diploid. So if you want to go back in the video, you can see there's 2 chromosomes. They're both replicated in the zygote. So that's diploid, 2 sets of chromosomes. But it says actually, if they're in the spore nucleus, we know that it's haploid. There's 7 chromosomes. So you could think of like 7 chromosomes like this, maybe all different sizes. There you go. So 7 chromosomes, or 7 chromatids as they are in the spore nucleus. So place a tick in the box next to the number that represents the number of chromatids present in the zygote. Well, the zygote has got 2 sets of chromosomes, so it's going to be double what we've just seen in the spore nucleus. But we know that each one is still replicated in the zygote. So let's have a look. So we've got, if I draw these out again, there you go. So these are replicated, okay? So there we can see there's actually 14 chromatids. However, this is only one set of chromosomes. They're just replicated. So we'd actually have the second set of chromosomes here. So we can see this is the second set of chromosomes because the zygote is diploid. And that will be another 14 chromatids. So if you count up all the chromatids from the zygote, so it's got double the number of chromosomes and each chromosome is replicated, it would actually come out as 28. So one of the misconceptions with this question was just going, oh, 7 chromosomes and each chromosome is a replicated chromosome with a 2 sister chromatids, so it must be 14. But there's double the number of chromosomes and they're all replicated in the zygote, as the diagram actually does show us. Okay, the scientists then recorded the arrangement of colour spores inside many narrow tubes. So they've collected 4 spores inside each of these little narrow tubes, and they've counted how many had this specific arrangement. So they found that there were 3 different arrangements of spores. It could be pink, pink, white, white, or it could be white, white, pink, pink, or we could have pink, white, alternative, pink, white, which is tube number 3. Now, if you look at the data, the numbers of tubes with pink, pink, white, white spores is 81. The number of narrow tubes with white, white, pink, pink spores in that arrangement, 78. These are quite similar numbers. So we might want to highlight that as we annotate the question. Hopefully then you can see that the number of narrow tubes with 4 spores in pink, then white, then pink, then white is actually a lot lower. So we'd annotate this and say that's actually quite a lot lower. There's no standard deviation bars, so we don't know if it's a significant decrease or not, but we do know that it looks a lot lower than 81 and 78. There's only 10 tubes with the type of arrangement that we find in tube number 3. So then it says, use all the info in the question. What can you conclude from the scientists' results about the movement of chromosomes in meiosis? So we need to think about what the key ideas about chromosomes in meiosis. Well, we know we get independent segregation. Now, that means a bivalent can attach to a spindle fibre like this. So I'll just put those on there. P. And then you've got the other one with the 2 alleles, that's white and that's white. Okay, or it can arrange the opposite way. So the 2 homologous chromosomes could be the white one on the left and the pink alleles on the right. So if we had the scenario at the top with this bivalent attached to the spindle fibre, we'd get 2 pink spores on the left and 2 pink spores on the white ultimately, which would equal tube number 1. If we had the arrangement here on the right, we'd get 2 white spores on the left and 2 pink spores on the right, which matches tube number 2. Now, because this is random, which way the bivalent attaches to the spindle fibre, we're probably going to get an equal number of these types of arrangements in the tubes. So half of the cells that undergo meiosis 1 will have the arrangement at the top for this bivalent. The other half of the cells, because it's fairly random, will have the opposite arrangement of the bivalent attached to the spindle fibre. So the bivalent can attach either way, either with the pink alleles on the left on that homologous chromosome, and the white alleles on the right on that second homologous chromosome, or it can be the opposite way. Now, the third tube here, we can see that, yes, we get fewer tubes with this arrangement of spores, but it's not two pink on the left and two white on the right. Instead, it's pink, white, pink, white. So what's going to happen here is, imagine you've got the first chromosome, and that's pink, pink, and you've got the homologous chromosome that is white, white. Imagine we had crossing over. So what you'll end up with is this bit of the chromatid crosses over with this bit. So because of crossing over between, or at the chiasmata point between these non-sister chromatids, we're going to end up with 4 spores, or 4 daughter cells. The first one with a pink allele, the second one not with the pink but with the white, because that was crossed over with the other chromatid. The third spore would have a pink allele, and the fourth spore would have a white allele because of the crossing over event in that bivalent. So we can see pink, white, which matches tube number 3. So let's just look at the answers then. Let me just see if I can get rid of the annotation. Okay, I'll just have to pop the answers over the top of what we've got. Just bear with me. Okay, so the answers then. Independent segregation occurs where homologous chromosomes in a bivalent are separated to opposite poles of the cell. That's independent segregation. And it's the random segregation of homologous chromosomes from the bivalent. So the homologous chromosome, which is made of 2 sister chromatids joined at the centromere on the left, that might be maternal. The matching homologous chromosome on the right of the bivalent, again made of 2 sister chromatids joined at the centromere, maybe the paternal homologous chromosome. That would end up in the right-hand side of the cell, and therefore the second cell at the end of meiosis one, or the bivalent could attach the opposite way. And we get an even distribution of alleles in tube number 1 as we get in tube number 2. It's about the same, very similar. We get 81 tubes with tube number 1 arrangement. We get 78 tubes with tube number 2 arrangement. So that even distribution indicates that independent segregation is random. So we're trying to use the numbers off the table. Now, we also mentioned crossing over. So we know crossing over in cells will take place in order to produce the arrangement of spores that we get in tube number 3. And we know because crossing over is rare, there's fewer tubes with that arrangement of pink, white. So there's only 10 tubes with that arrangement. So crossing over does not occur in every cell that undergoes meiosis 1. It occurs in about 10% of cells that undergo meiosis 1. So 90% of the cells wouldn't have any crossing over. So the pink and the white alleles would not be swapped over between homologous chromosomes. That would only occur in about 10%. That's why the third arrangement in tube number 3 is actually, the numbers that we see of that is a lot less. It's only about 10 tubes compared to 81 or 78. Okay, so then it says meiosis results in cells that have haploid numbers of chromosomes. That means one set of chromosomes, and show genetic variation or genetic uniqueness. And then it says explain how. And this is like a 6 marker, so there's quite a lot of information that we need to write down for this, but it's the same 2 ideas really, about independent segregation and about crossing over. Now, crossing over actually occurs first in prophase 1. So as soon as the bivalent forms, we might get, even though it's quite rare, we might get crossing over between non-sister chromatids between the 2 homologous chromosomes in the bivalent. So the replicated chromosomes, 1 maternal, 2 sister chromatids joined at the centromere. The second homologous chromosome, paternal, again made of 2 sister chromatids joined at the centromere. They pair up through synapsis to form the bivalent. That's in prophase 1. We can see these replicated chromosomes because they're super condensed. We might get crossing over at the chiasmata point when equal length of chromatids are swapped over. And that's because it is quite rare, doesn't occur that often, but it does, if it does occur, it does produce new combinations of alleles. So we saw before the pink allele on one chromatid was swapped with the white allele on the chromatid, one of the chromatids of the other homologous chromosome. Now, homologous chromosomes separate randomly from the bivalent. Now, the bivalent can attach one of two ways to the spindle fibre. The maternal chromosome could be on the left and the paternal one on the right, or the paternal chromosome could be on the left and the maternal one could be on the right. So that's fairly random which way the bivalent attaches to the spindle fibre. And that's called independent segregation. Now, we know the separation of homologous chromosomes is random to both poles, depending on which orientation the bivalent attaches to the spindle fibre. But again, that produces new combinations of alleles because it's random which chromosome ends up in which daughter cell. So in this mark scheme, the idea of new combinations of alleles was credited only once. So crossing over can produce new combinations of alleles, as can independent segregation of the homologous chromosomes from the bivalent. Now, we know in Meiosis 2, because the question is actually asking us about the haploid number of chromosomes. So we got to reference that, as well as genetic variability, which we've already talked about. So homologous chromosomes in Meiosis 1 are separated from the bivalents, and then in Meiosis 2, each replicated chromosome, which is still made of two sister chromatids joined at the centromere, the centromere now splits and the sister chromatids get pulled to opposite pole to the cell in a V shape. So the gamete cells, or the spores in this case, that get formed will have one chromatid or one sister chromatid in them from each replicated chromosome. So not only is the DNA mass halving, but also we know the gametes or the spores will be haploid because they only have one set of chromosomes. Okay, so another question then, it says nucleus A and nucleus B come from the same organism. And then the diagram shows these nuclei immediately before either mitosis or meiosis is what this question really means. So think of the nuclear division as being either mitosis or meiosis. The nuclei formed immediately and the nuclei formed immediately after their division. The table gives information about some of the nuclei shown in the diagram. So it's saying the number of chromosomes and the mass of the DNA. So if we look at nucleus A, okay, we know this is a somatic cell. It's probably a stem cell because it is dividing by mitosis, and you've got stem cells in every tissue of your body. We know the DNA would replicate after S phase, and it's saying there's 8 chromosomes. So just before mitosis begins, which is actually after S phase, so we put S phase there, okay, we know there's 8 chromosomes, and all 8 chromosomes are made of 2 sister chromatids joined at the centromere because it's after DNA replication, and it's telling us that there's an arbitrary unit of 600. So if I drew out the 8 chromosomes, they'd look something like this. That's 1, 2, 3, 4, 5, 6, 7, 8 chromosomes, all replicated, made of 2 sister chromatids joined at the centromere. So that equals 600 arbitrary units in the mass of the DNA. Now it says nucleus B, because it's from the same organism, nucleus B is also diploid, just like nucleus A would have been diploid. So there's 8 chromosomes, and it's saying also just before meiosis begins, which is after S phase, remember that's in interphase, it's the same 8 chromosomes and the mass is 600. Then it's saying what would the number of chromosomes be in nucleus C? Well, we know mitosis produces genetically identical daughter cells. So we could say genetically identical. that means the ploidy is the same, i.e. is it diploid, haploid? Well, it must be diploid. The chromosome number is the same. Okay, but let's think carefully about the mass of the DNA. So yes, there would be 8 chromosomes in nucleus C, but they wouldn't be replicated chromosomes like we've got here. They'd only be made of 1 sister chromatid because the sister chromatids were separated during mitosis. So actually the number of chromosomes is going to be 8. So I'll put the answers on the left and then we'll check them in a minute. But the mass of the DNA, because they're not made of 2 sister chromatids like this, they're only made of 1, the mass is going to be 300, which is half of the one above it. There you go. Now, nucleus D then, so we'll pick this one down here. Number of chromosomes, okay, so Meiosis 1 and Meiosis 2 would have taken place here, Meiosis 1 and Meiosis 2. So after Meiosis 1, there would be a haploid number of chromosomes, that would be 8, sorry, that'd be 4. And then obviously the 4 replicated chromosomes would split during Meiosis 2. So in nucleus D, you would have 4 chromosomes, and each would be basically a single sister chromatid. So it must be 4 single sister chromatids in nucleus D, and therefore we're going to go for half the mass compared to nucleus C. So if we just check those answers, 8 chromosomes in nucleus C. They're all sister chromatids. They're not 2 sister chromatids joined together. So the mass of the DNA is half. So 600 divided by 2 is 300. Nucleus D, again, we've got a haploid nucleus because it's meiosis, so a haploid nucleus. So it's half the number of chromosomes, which is 4, and they're all single sister chromatids. So therefore, it must be half of nucleus C. So it's 300 divided by 2 is 150, and that's what we end up with. Okay, so I hope that video was useful for you. If you think it was, please do subscribe or do hit the like button and give us some feedback in the comments below. Okay, thank you.